Welcome everybody to today's broadcast. This is Susan Hedstrom from the Foundation for Prader-Willi Research, and joining us today for our presentation is Dr. Eric Hollander from Montefiore. This is the University Hospital for Albert Einstein College of Medicine. Dr. Hollander will be talking to us about intranasal oxytocin. This is a study that he is currently running, funded by the FDA. Without further, one last thing, we will be answering questions at the end of the webinar. So if you do have questions, you can use the control panel to submit any questions. We will collect those, and when Dr. Hollander is finished with his presentation, we will run through those questions. So without further ado, I give you Dr. Hollander to present to you on oxytocin.
Okay, well, thanks very much, Susan. It's really a pleasure to be here. And I'm grateful to the Foundation for Prader-Willi Research for all of the help that they have given us over the years in terms of our studies in Prader-Willi syndrome, and in being able to speak with you about this particular study today as well. So I'm at Albert Einstein College of Medicine and Montefiore Medical Center in New York City, and I'm going to be talking about the study on intranasal oxytocin versus placebo for the treatment of hyperphagia in children and adolescents with Prader-Willi syndrome. And this is currently a grant that's funded by the Orphan Products Division of the Food and Drug Administration, and where we're actively recruiting 50 children and adolescents with PWS. My contact email, by the way, for you is ehollander@montefiore.org. So our program focuses on Prader-Willi syndrome and also on autism and other obsessive compulsive spectrum disorders. And our overall approach is to develop new therapeutics that target different core symptom domains that may occur in Prader-Willi syndrome and can also be expressed across other conditions as well. And our understanding is really what are some of the underlying brain mechanisms that play a role in modulating these clinical symptoms, and then matching particular treatments to the underlying domain in order to get the biggest impact or the best improvement. We're also involved in a study with CBDV, cannabidivarin, which is one of the new cannabinoid type compounds. And I'm going to be doing a webinar on this next week. And we're involved in a study with CBDV in Prader-Willi syndrome, which is funded by the Foundation for Prader-Willi Research. And we're also working with GW Pharma that's extracted this particular compound. That is a study where we're looking at the effects of cannabinoids on irritability, also on repetitive behaviors and rigidity, and on problems in terms of social cognition as well, or back and forth type of communication. Today I'm going to be talking about our study with oxytocin, and in particular where we're going to be targeting the hyperphagia as well as some of the rigid and repetitive behaviors as well. And we're using an intranasal oxytocin. One of the points that I would make, or one common theme, whether it's a treatment study looking at CBDV or cannabinoids or intranasal oxytocin, is that both of these compounds seem to have some kind of effect on the balance between excitation and inhibition. If excitatory pathways in the brain are too active, then you can have too much excitation. If you have too much excitation or not enough inhibition, then you can have disruptive behaviors or explosive behaviors. You might have repetitive behaviors or perseverative behaviors. You can also have things like seizures, for example, if there's too much excitation in the brain. And we believe that both oxytocin and the CBDV partly may exert their effects by modulating this imbalance between excitation and inhibition. The other thing that I think is important is that both of these treatments may have some effects on dampening down the immune or inflammatory response. And, in issues around obesity, for example, or Prader-Willi or other developmental problems, one of the things that's been seen is that these inflammatory pathways may be turned on. Oxytocin, for example, can dampen down that immune inflammatory response. Another thing that we've been looking at in Prader-Willi syndrome is looking at issues around rigidity. And in particular, the idea that there can be a behavioral rigidity, so people can experience discomfort if there are changes in the environment, if there are changes in the timing of meals, or changes in routine, for example. That's behavioral rigidity. Or there can be a cognitive rigidity. People may see things a particular way and have a hard time transitioning from one set of thinking to another, or being more adaptable. And we've developed this measure, the Montefiore-Einstein rigidity scale for Prader-Willi syndrome. And we're also looking at as an outcome in our different treatment studies, and then trying to understand whether or not that rigidity domain goes along with certain genes as well. If I could have the next slide, please, Susan. Okay, so one question is, why are we interested in looking at oxytocin in Prader-Willi syndrome? So one of the reasons is that it's been shown in the brains of individuals with Prader-Willi syndrome that the number of oxytocin neurons and the size of these oxytocin producing neurons in the brain may be reduced. So they may have fewer or smaller oxytocin neurons in the hypothalamus, for example, going to the posterior pituitary, and then those systems, secrete oxytocin into the bloodstream as well. In mice, if you reduce these oxytocin neurons, then mice tend to overeat to the point of severe obesity. In those mice where there's a reduction in oxytocin neurons, but we can administer oxytocin, we can reduce that obesity or the compulsive eating or hyperphagia. So giving oxytocin in animals will decrease the food intake and decrease the body weight of those animals. So targeting this pathway makes some sense. It's also been shown in other conditions like autism spectrum disorder that administering oxytocin may help with certain behaviors, social behaviors, eye gaze, the ability to recognize emotions in language, for example, and to develop trust with other individuals as well. And that administering oxytocin may decrease certain repetitive behaviors as well. So there's some evidence in animals that the oxytocin system may be involved, that administering oxytocin may decrease obesity or improve body weight. And in patients with autism, for example, there's some evidence that administering oxytocin may help with social communication or may improve certain repetitive behaviors as well. If I could have the next slide, please. So there have been some clinical trials of oxytocin in Prader-Willi syndrome. The studies have been relatively few, and the results have been somewhat mixed. So there have been some positive findings, there have been some negative findings, and there have been some inconclusive findings. One is that there are big differences in terms of the methods, between these different studies. So for example, some people have studied the effect after a single dose or after a week of treatment. Other studies have looked at 8 weeks of treatment. There have big differences in terms of the sample size. Some of the studies have been very small, others have been somewhat larger. There have been big doses in terms of the dosing. we don't know exactly what the optimal dose is. There was one study that used relatively high doses of intranasal oxytocin. And in some of those individuals at these high doses, there was a little bit more aggression or temper tantrums. One idea is that perhaps by administering high doses, you may be getting some binding of the oxytocin to other receptors, like the vasopressin 1A receptor, for example. And then, one important issue is outcome measures. Like, how do you measure these kind of changes in different populations? Well, you can measure weight, or you can measure body mass index, or BMI, but then you can measure things like hyperphagia, and there are different scales for that, or you can measure rigidity, and there are different scales for that. Or there may be other, what are called like biomarkers, so other kinds of things that you can measure directly that may not relate to behaviors, for example. You can look at outcomes that are based on what the caregivers rate, or you can look at some measures that are measured by a clinician, or you can look at other measures that are directly measurable, so like a biomarker type measures as well. And that differences in all of those kind of issues may partly explain why you're getting different responses in different studies. Next slide. So here are some of the outcome measures that we're looking at in this current study, which is funded by the Orphan Products Division of the Food and Drug Administration. Our primary outcome is hyperphagia, and our primary outcome measure is this hyperphagia questionnaire, which has been sort of modified and scaled, and where there have been some use of this particular measure in some clinical trials in Prader-Willi syndrome and in other conditions as well. So that's our primary measure, is what's the compulsive drive to eat, or the hyperphagia, measured by this particular hyperphagia questionnaire that's been scaled. The secondary measures that we're looking at, one has to do with repetitive behaviors and rigidity. And we're using this, the RBSR, the Repetitive Behavior Scale, which has a number of subscales. And then we're also looking at this rigidity, and we're looking at both rigidity in terms of changes in the environment, rigidity in terms of the cognitive rigidity, and then how much people protest when, if there's a change in the environment, people experience discomfort and then protest. And to do that, we're using the Montefiore-Einstein rigidity scale, the Prader-Willi version of that. We're also looking at what impact these measures are having on overall quality of life. So we have a couple of scales, this World Health Organization Quality of Life Scale measure, or the Caregiver Strain Questionnaire. Another scale that we're looking at is overall irritability or disruptive behaviors. And the irritability and disruptive behaviors may be relating to things like temper tantrums or self-injury. And we're using the aberrant behavior checklist irritability subscale. Finally, we're looking at salivary concentrations. So the concentrations of oxytocin that can be measured in the saliva. So we're taking samples there. And then we're looking at some direct measures like weight, body mass index, and body composition measured by this bioelectrical impedance analysis. And then finally, we're looking at a global improvement measured by the Clinician Global Impression Improvement Scale. So how much overall the clinician feels is the individual very much improved, minimally improved, unchanged, minimally worse, much worse, for example. Next slide. So there have been some studies that have looked at potential side effects of intranasal oxytocin, both in Prader-Willi syndrome and in other conditions like autism spectrum disorder or other conditions. Some subjects experience an increase in a sense of calmness or euphoria. Some individuals have noted an increase in energy. Some subjects have noted a little bit of lightheadedness. Some individuals have noted a little bit of drowsiness or fatigue, headache. Some people get a little bit of nasal irritation associated with the nasal spray. Some individuals have noted some dry mouth or dry throat. And I mentioned that there was one study that used at higher doses, 40 international units of oxytocin, where there was an increase in temper tantrums or aggression in adults who received that. Next slide. So in terms of safety assessments, well, one of the things that we're looking at is safety labs. So we're looking at things like sodium levels or liver functions, kidney function, blood count, things like that that can be measured in routine blood work. We're looking at other measures like weight, for example, and height and things like that. We have formed an independent data safety monitoring committee, or a DSMC, and this is a group of individuals who are able to start to look at the data as it's coming back and see whether there are any trends or issues that are coming up in terms of safety. The study is a placebo-controlled trial, so the investigators don't know whether the any individual subject is getting the active oxytocin or whether they're getting a placebo or like a sugar pill or sugar spray, for example. The independent data safety monitoring committee, if they want to, can break the blind, for example, to look at these trends if there is something that's come up that's of some concern. And then the study doctor at each of the study visits, talks with the family and discusses any concerns or any issues relating to possible adverse events as well. Next slide. So the design of the study is that subjects will come in for a period of 8 weeks where they'll participate in this double-blind or placebo-controlled trial. So they're either getting the active intranasal oxytocin or they're getting a saline kind of a nasal spray placebo. There is a total of 5 visits that occur here at Albert Einstein College of Medicine, and patients may also come into the Clinical Research Center here. We're recruiting for a total of 50 children or adolescents between the ages of 5 to 17. And then we have the ability to do some dose optimization. So we start at 16 international units in a nasal spray. If we see that there are no side effects and we see that there's no benefit, then we have the ability to titrate up on the dose to 24 international units. If we think that individuals might possibly be having some kind of side effects, we have the ability to lower the dose as well. So we can optimize the dose individually. Next slide. One issue is that the travel costs for people to come here from different sites can be reimbursed, and that can include airfare and a hotel. Again, we're seeking 50 subjects. We're reaching out to people all across the country. And then, if there are travel costs, we can reimburse those travel costs as well. Next slide. So here is the primary inclusion criteria. Individuals need to have a diagnosis of Prader-Willi syndrome. They have to be between age 5 and 17 years of age. They have to be in this nutritional phase 2B or 3. Basically what that means is that they need to have hyperphagia or compulsive eating. They have to have at least a moderately severe sort of hyperphagia or compulsive eating on the hyperphagia questionnaire. We like people to be on a stable dose of growth hormone for about 3 months before being randomized. So everybody will be on growth hormone and then have the oxytocin added. And we want them to be on stable doses of any other hormones as well, so that they can if they're getting testosterone or estrogen supplements, for example, we want them to be on a stable dose of that for 4 weeks prior to randomization, and then stay on that same dose throughout the duration of the study. Also, any other metabolic treatments that they may be getting, we want them to be on a stable dose. Anything that may affect appetite for 4 weeks prior to the randomization and during the study. And we want them to be on stable, any kind of medication, dietary interventions, or other kinds of behavioral interventions. We want that to be constant before and during the study. And then we also want the physical exam and laboratory tests to be normal in our Prader-Willi subjects. Next slide. So what are the primary exclusion criteria to be excluded from the study? Well, if they've been on any other kind of investigational agent, so something that's a non-approved agent in a research study for 1 month prior to coming in for randomization, we want all of the children to be on a stable dose of growth hormone. So if they haven't been on the growth hormone, then we want them to be on a stable dose of the growth hormone before randomizing them. If the kid weighs less than 40 pounds, then they may not be eligible. If they have unstable type 2 diabetes, so that they're medically unstable and then they need other kinds of interventions, then they might not be a good candidate. They can have other medical comorbidity, but if it's unstable, and they may need other kinds of interventions, then they may not be eligible for the study. They can have another concomitant psychiatric disorder, but that the Prader-Willi syndrome needs to be the primary problem. They could have a mood disorder or an anxiety disorder or a developmental disorder or a history of drug abuse, but we want the Prader-Willi diagnosis to be the primary diagnosis. And then any unstable medical condition that would interfere with the ability of somebody to participate or stay in the trial. And people who have marked abnormalities on blood tests or on EKG or other physical abnormalities, that may make them ineligible as well. Next slide. And if people are interested in participating in the trial, then they'd like we'd like them to contact our study psychologist, Bonnie Taylor, and her telephone number is 718-839-7530, and her email is botaylor@montefiore.org. So the phone number is 718-839-7530 or botaylor@montefiore.org. And I believe that's the last slide.
Correct.
Okay, so maybe we could just leave this up, and then I would be glad to take any questions or respond to any comments as well. So let me know if anybody has any questions.
Wonderful. Thank you so much, Dr. Hollander. For those of you who are on the line with us, you are welcome to submit any questions you have. You have a control panel open for GoToWebinar. If you type in the questions and push and enter or submit, they will come to me and I can present them to Dr. Hollander. I have a couple of questions in the queue, so we can go ahead and start with these. Dr. Hollander, could you go into a little detail on how oxytocin is different from carbetocin?
Right, so oxytocin is a naturally occurring peptide. It's made in the hypothalamus, and then it's secreted into the bloodstream when it's secreted from the pituitary gland, the posterior pituitary. So our brains make oxytocin. Oxytocin can also be administered through a nasal spray. Carbetocin is a modification of the peptide oxytocin, where one of the amino acids is changed. So it's not the naturally occurring substance that's found in individuals' brains and in their bloodstream. It's a synthetic compound which differs by one particular amino acid. You know, one reason that carbetocin was created was that the carbetocin, when you modify the naturally occurring compound, there is a slight longer half-life in the plasma. So it stays in the plasma a little bit longer. That was an issue because when carbetocin used to be administered, let's say by an intramuscular injection, so in areas like rural Africa, if women were having a hard time progressing in terms of labor and delivery, then you could have a doctor out there in the bush administer this synthetic form of oxytocin, the carbetocin, by an intramuscular injection, and it might stay around in the bloodstream a little bit longer. I don't think that there's any particular advantage, for example, of modifying the oxytocin peptide to a synthetic form or modifying this amino acid to create carbetocin. We know that the oxytocin seems to get into the plasma and gets into the spinal fluid when it's administered in an intranasal form. And this has been shown, for example, in studies of rhesus monkeys, for example. It turns out that the clinical effects that we see often relate to the oxytocin binding to these oxytocin receptors that are in the social brain. And administering the oxytocin through an intranasal fashion will, some of that peptide will bind to these oxytocin receptors and then influence things like appetite, for example, or might affect things like rigidity through their binding to oxytocin receptors. There is a study using the carbetocin, again, this synthetic form of that, and the difference is that one of the amino acids in the naturally occurring peptide has been modified.
Wonderful. Would you expect carbetocin and oxytocin to work similarly, or would you expect one to be better than the other?
Well, I would expect that the oxytocin might, given that it's the naturally occurring substance that actually binds to these oxytocin receptors, that there could be some natural advantages to using the naturally occurring compound as opposed to a synthetic or modified compound. As I mentioned, one advantage of the carbetocin is that it seems to have a slightly longer half-life, so it stays around in the plasma a little bit longer. But the amount of time that it spends in the plasma doesn't seem to be related to any of the clinical effects, which seem to be more related to the binding of the compounds to the oxytocin receptors in the brain. So I don't see any particular advantage to modifying the peptide.
Wonderful. So you mentioned that there were, I believe, 5 study visits associated with this particular study. How long would one expect a typical visit to last?
Well, that's a good question. So the first visit is a longer visit. The children come in, they get a diagnostic evaluation. They have some what we call structured interviews, where they go through certain kind of questionnaires. The clinician may ask some of the questions, and then also the clinician may ask the parent some of the questions, and then they may have a physical exam and get blood work and things like that. So the initial visit is a longer visit and could last an hour or two, I think, And then the subsequent visits are somewhat shorter than that.
Logistically speaking, if someone was to fly in to participate to this study, would they need to come in the night before or stay the day of? How long do you anticipate their trip would need to be?
Well, they wouldn't have to come in the night before. I guess it really depends on what is most convenient and how the flight schedule works out. I mean, so we'd want the person to come in the morning and then be available, you know, for the day of evaluation for at least a couple of hours or so. So I guess it kind of is around just scheduling and convenience. I mean, if someone is going to be flying from the West Coast, then they probably, you know, they might want to come in the night before and settle in and then come in first thing in the morning.
Okay. Will all patients who participate have a chance to receive oxytocin?
No, I mean, the study is a placebo-controlled trial, so they may get the active treatment or they may get the placebo treatment. You know, everybody will get the advantage of the physical exam and the blood work and careful assessment. We'll share all of that information as well. But in this particular study, there's not a open label maintenance kind of an arm. So not everybody is guaranteed to get the active treatment.
Okay. And after the study concludes, will anyone have continued access to oxytocin?
Well, so some people can get access to oxytocin in that they can, if they have a clinician who's comfortable with the administration of that, and they have a pharmacist who's a formulation pharmacist, and they know like how to administer it and what to look for, then it's possible that they could get a sort of access to that treatment. I mean, there's a big advantage in terms of getting it in a clinical trial. One is it's free. You don't have to pay for out-of-pocket costs for medicines that may need to be formulated and then that are not reimbursed by insurance, for example. And then you'd have careful follow-up. We'd be able to do systematic scales to see whether or not, you know, some of these behaviors, whether it's the hyperphagia or the rigidity or the repetitive behaviors or some of these other type measures, whether or not there's any kind of systematic improvement or not. So careful observation and good workup.
Right. We still have a couple questions in the queue. I just want to remind everyone who's on the line with us that you're welcome to submit questions. We have about 5 minutes left. So if you have any questions on your mind, please feel free to submit them and we will make sure that they get answered. Next question, why do you require participants to be on growth hormone?
Well, that was an interesting question, and we had consulted other experts and then had our study is funded by the Orphan Products Division of the Food and Drug Administration, and as part of the whole process, they had outside scientific reviewers as well. One potential source of variation is if some people were getting growth hormone and other people were not getting growth hormone, that it's possible that some of the measures that we're looking at could potentially be influenced by whether or not they're getting growth hormone. So in order to kind of standardize the treatments and decrease variation from subject to subject, you know, it was concluded that it might be best to have that be constant among everybody who is participating in the trial, and that reduces one of the variables that could, you know, reflect different effects by different patients. So that's one variable that's sort of going to be constant among everybody. The other issue is that, you know, it's also conceivable that you could have an interaction effect. So you can have a synergistic or greater effect in those individuals who are getting both growth hormone and the oxytocin. And it's possible that oxytocin and growth hormone may have sort of additive or synergistic effects as well.
Is there evidence from previous studies that may support that or indicate that?
There are some suggestions that that may be a potential source of variability from person to person, but there are some suggestions. So it's possible, like for example, people who happen to be on stimulants and oxytocin may have a particularly good response. So that's been seen with some other things, that there could be potential additive effects between different treatments. And then it's interesting that, you know, growth hormone can work on certain second messenger pathways or molecular pathways that oxytocin works on as well. So from a hypothetical standpoint, on understanding how they can work on certain cell cycle pathways, there could potentially be additive effects.
Wonderful. How did you select the age range that you are currently studying? Is there a reason why you stopped the eligibility at 17?
So that's a good question. And, you know, one of the things that's a little bit different when I talk to you next week about our study with this cannabinoid compound called cannabidivarin, or CBDV, that's a study that we're looking at both in children and in adults. And in that particular study, we're going up to the age of 30. One of the issues here in terms of figuring out the age range is that in looking at some of the measures that we're using, those measures have been sort of standardized in child populations. And if we would study the adults and children together, it's possible that there may be some differences in terms of how adults respond to some of the outcome measures or the ratings, where there could be some variability in differences in adults and children that might confuse some of the findings. So again, we wanted to get a more homogeneous population, which is children and adolescents.
Sure. Well, thank you, Dr. Hollander, for joining us today and sharing details on your oxytocin study. I would like to invite all of you, here are the details, to our next webinar, which is going to be Monday, March 2nd. That's this coming Monday, where Dr. Hollander will be sharing details on another study he's undertaking. This is the CBDV study. The study will be opening soon and will be enrolling patients ages 5 to 30. So if your child is within those ages, please join us next week to learn more about this study. If you do have any further questions, Dr. Hollander did provide you with contact information, and you are encouraged, of course, to reach out to his study team to answer any questions you may have about eligibility and participation. Thank you so much for joining us, Dr. Hollander, and thank you so much to the community for joining in as well. Please continue to share this opportunity with those who you think might be interested. Have a great day.